This is the instruction video for the DNA foldable. The first step that you're going to want to do for this foldable is to create the foldable, which means you're going to have to do a couple of folds. The first fold you're going to do is you're going to want on this line to fold the foldable inwards. So I'm going to fold on this line the foldable inwards. All the way down. Going to go ahead and open that back up after you've gotten a good crease on this line. And you're going to do the same thing on this line here. So you're going to fold it inwards on that line. And you're going to open it back up. Those are the first two folds. Now we have a couple of more. So go back and fold this guy back down. And now you're going to fold this halfway. You can see that there's a line here. We're going to fold on this line, but we're going to fold it backwards. So go ahead and fold. Backwards on that line. You're going to do the same thing with this side. So go ahead and fold it in, like before. And then you're going to fold this one backwards on that line. So your foldable should look like this. Now we have our DNA molecule. As you can see here, we have two sides of the DNA molecule. One side of the DNA molecule already has the bases labeled. On this opposing side, you're going to go ahead and label the complementary base, the base that would be attached to the base on the left. So here, for example, you will see A will match with T, if you remember Chargaff's rules of pairing. C will match with G, and you're going to continue all the way down the strand finding the complementary bases. So at this point, go ahead and pause the video until you have all of the pairs labeled all the way down the foldable. Okay, we're going to continue. So from here, we're going to try to replicate this DNA. So you have a full DNA strand here. These should all be labeled. Now we're going to replicate the DNA. And the first stage of DNA replication is unwinding and unzipping the DNA with DNA helicase. And so we already have the DNA unwound. It's not in a spiral right now, right? It's flat. And then DNA helicase is going to come through and unzip the DNA molecule to pull these apart from one another. So go ahead and pull these apart and open up your paper all the way. And you should have two labeled strands on the outside, but these ones on the inside are not labeled. So go ahead and open. Now we're going to act as DNA polymerase and go through and add the pairs to the strand of each individual nucleotide, just like that enzyme will come through and add each individual nucleotide on here. So for example, A is going to go to T, G is going to go to C. You're going to continue that all the way down the strand. But you're also going to do this side. This side should be completely labeled already. So T is going to pair with A, and G is going to pair with C, and then the rest of them you're going to label down on that strand. So you're going to act as DNA polymerase and add the corresponding nucleotides onto each of these strands. Go ahead and pause the video until you have all of the nucleotides the nitrogenous bases labeled. Now the next thing we're going to do, all of the nitrogenous bases should be labeled. Now we're going to label the deoxyribose sugars. So I'm going to put next to deoxyribose here, because it asks you to label, I'm going to put an S here and circle it. I'm going to label all of my deoxyribose sugars with an S. Now, remember which one of these is the sugar. The sugar is the 5-sided pentagram here. And so you're going to label all of the pentagrams on both sides of both DNA molecules with the sugar. Go ahead and pause and complete labeling all of the sugars. Now we are going to label the phosphate groups. So go ahead, I'm going to put a P to show that I'm labeling the phosphates with a P. And now if you remember, the phosphates are on the backbone of the DNA molecule. We've already labeled all of the sugars. Now we're going to label the phosphates. So the phosphates are these small circles here. So you're going to label all the phosphates down all sides of both DNA molecules. Go ahead and pause the video until you have all of the phosphates labeled. Next, we're going to label the nitrogenous bases. So go ahead and find the nitrogenous base and label the nitrogenous bases. So all of these on both sides in the middle are going to be nitrogenous bases. So I'm actually going to circle or make a square around a nitrogenous base. I'm going to draw an arrow to label it nitrogenous base. Next, I'm going to label the backbone of the DNA molecule. So the backbone is the phosphate sugar backbone here. So we are going to label the backbone here. Now our backbone is labeled. Next, we need to label a nucleotide. If you can remember, there are 3 portions of a nucleotide. We have a deoxyribose sugar, we have a phosphate, and a nitrogenous base. So you're going to find somewhere, anywhere on the molecules, one nucleotide. So I'm going to choose this nucleotide here. I'm going to draw a square around it, around all 3 of those pieces, the phosphate, the sugar, and the base, and I'm going to label that nucleotide. Now we're going to label the original parental strand. So if you remember, the parent strand is the first strands that we start off with, the ones that we use as a template. So these 2 outer strands are going to be our parent strands because they're the old strands. So you can go ahead and label these strands the parent strands. And then we're going to label the complementary strands. The complementary strands are going to be the new strands that are complementary to the parents. So go ahead and label the complementary strands. Now before you turn this in, you're going to finish by answering the question, what role do the following enzymes have in DNA replication? So you're going to answer this question here, about these two enzymes. And you're also going to write the steps of DNA replication here. This is going to serve as a sheet for you to reference during the unit. That way at any time, if you forget what a deoxyribose sugar is, you can go back and reference it. If you forget what a complementary strand is, you can go back and reference it as well. If you needed to remember how to do base pairing, you're going to be able to see how you did that there, and it's going to be a good resource for you to use to study this unit. When you're finished, you're going to take a photo of your complete diagram, and you're going to upload that into the assignment in Canvas where you got this sheet to print off.